Okay, the lysosomes, another organelle in the cell, are considered the killing body of the cell. These organelles here are ones that are responsible for destruction when it comes to cell activity. The lysosome is made from the ER and the Golgi, so it is actually one of the products coming from the Golgi, so it's made from the rough ER. The lysosome is a membrane sac of hydrolytic enzymes, acid hydrolases. What is an acid hydrolase? ASE enzyme, insert water. Remember, if you insert water, you split stuff apart. But to activate the enzyme, it must be in an acidic environment. To activate the enzyme, it must be in an acidic environment. So that means the cell generally keeps this in a basic environment. Okay, for example, police officer carrying a gun, but he has a safety on the gun and a snap over his gun, so he doesn't shoot himself in the foot. So you don't want these enzymes to activate in a cell because it would destroy the cell. So you keep it in a basic environment until it's needed, and then you start making it become acidic. Lysosome enzymes can hydrolyze, break apart proteins, fats, polysaccharides, nucleic acids. So if a bacteria gets in, the white blood cells could secrete lysosomal enzymes to get rid of it, digest bacteria, and so forth. It also gets rid of non-functional organelles. You mark them of destruction. That means inside a cell, just let's say a couple of organelles go bad, you don't have to necessarily throw the whole cell away. You can just get rid of those particular organelles. Can release glycogen, break down and release glycogen, and so forth. Some types of cells can engulf another cell, phagocytosis. We talked about that. So when you undergo phagocytosis, then the lysosome could come and fuse with that phagocytotic vesicle and try to destroy what's in there. Okay. Digestive enzymes showing you there, cell membrane. So now we're looking at, let me go to this, terms for cell destruction. That's what this is showing, terms for cell destruction. Autophagy, heterophagy, apoptosis, necrosis, and autolysis. So let's look at these terms. Autophagy is a catabolic process involving the degradation of cells' own components due to lysosome machinery. Okay, what is this talking about? This means that inside the cell, a couple of organelles go bad. You go ahead and take those organelles out, self-eating within it. Heterophagy. This would be when, like with phagocytosis, you took something in, engulfed it, because hetero means other than, and then you destroy it. So this would be destroying some of the organelles in the cell that are no longer needed. You can't destroy some organelles. You can't destroy the nucleus or you wouldn't have a cell. But those that you have a multitude of, redundancy, you could destroy some if they went bad. Heterophagy, taken into the cell, phagocytosis or peno, and then you destroyed inside. So you took this from the outside. Okay? I won't get into ubiquitin. Apoptosis, big term. I want you to know it. Programmed cell death. That means in the genes, there is a destruction signal that says this cell needs to die. For example, when you're developing inside your mom's womb, you develop hand plates and feet plates. You don't have separate fingers and toes. Then at a certain time, based in the genetics, the cells between the fingers, the skin between the fingers, those cells will start to die and separate the fingers. Sometimes, like in Down syndrome, you sometimes can get webbing, which means it didn't complete its process. So apoptosis is called programmed cell death. Programmed cell death. Necrosis is non-programmed cell death. For example, if you cut the blood supply off to something, it's going to die. It wasn't programmed, it's going to die. And that's what we call necrosis. I'm going to call that an unprogrammed cell death. Autolysis is where the whole cell itself gets destroyed. This is in contrast to apoptosis, where the cell does get destroyed, but it's programmed cell death. So let's go back. Autophagy, get rid of bad internal organelles. Heterophagy, phagocytosis, destroying what you brought in. Apoptosis, in the DNA, set certain time for the cells to die. Necrosis, unintentional. Necrosis, unintentional, and autolysis is a condition by where the cell is unprogrammed and dies. So that's what we're looking at. All right. We now go to the peroxisome, body of peroxide. The peroxisome is an internal organelle, and what it has is some peroxidase. Okay, so peroxidase is an enzyme. So listen to what I'm going to say. If doing chemical processes in the cell, some byproducts will be made. Oxygen will be released. This is kind of like a Dow chemical, and you're having waste products from your manufacturing. So inside the cell, due to normal chemical reactions, free oxygens will be released. Free hydrogens will be released. Free oxygens will be released. Free hydrogens will be released. Now, listen what I say. Free hydrogens, the atom oxygen, not O2, what we breathe in. That's a molecule. Free oxygens running around inside a cell could start and become a free radical. Free oxygens inside the cell could become a free radical. Free hydrogens could lose their electron and become a proton. Go back to earlier stuff. An H plus free proton would mess up acid-base balance because a free H plus is an acid. But these things are being released. So therefore, inside the peroxisome is an enzyme called the peroxidase enzyme. The peroxidase enzyme. What the peroxidase enzyme can do is take 2 free hydrogens and 2 free oxygens and make it into something called H2O2, hydrogen peroxide. Hydrogen peroxide is that stuff you get in the brown bottle at the drugstore. Now, hydrogen peroxide is far less toxic than having free oxygens running around. They could become free radicals and free hydrogens, which could become acidic. So H2O2 is formed because of the peroxidase enzyme. Now, what will happen is that when the concentration of H2O2 starts getting too high, it could be a little harmful itself. So you have another enzyme called the catalase enzyme. And what the catalase enzyme does is to take H2O2 and convert it into H2O and O2. H2O and O2. H2O and O2. H2O being water, O2 being oxygen, but the molecule. So other words, a major function is to break down fatty acids in the process of bake. So what can happen while it's doing its job, you can use some of those oxidizing substances also to break down fatty acid molecules. So the peroxisome serves many functions, but one major function is to to help with the elimination of some free radicals, and also in the breakdown of some of your fatty acid molecules. That's the peroxisome. Okay. The next up is the mitochondrion, and I'm going to actually do a separate video on the mitochondrion.